Okay, drug resistance. So what does resistance mean? So resistance means that antibiotics are not going to work anymore. So basically it means that the bacteria have found a workaround or a way so that they've developed a way so that the antibiotics don't kill them. So I want you to think about the difference between bacteriostatic and bactericidal again. So if we draw the graph, right, bactericidal, they're going to die. Okay, but if something is bacteriostatic, right, it keeps it flat. So basically, if we don't kill something off, okay, so if we give it antibiotic and we don't kill something off, the next time somebody gets sick, it's been exposed to that antibiotic and it's probably developed some sort of resistance to the antibiotic, maybe, okay? So because they adapt and they can survive the antibiotic. So the acquisition, how it happens, is mainly by genetic changes. Sometimes it's not genetic. It can be just mutations and natural selection. So the antibiotics, if the antibiotics are there, they can favor, the environment favors the mutants or the ones that have been changed. Okay, sometimes there's chromosomal resistance, and sometimes it's with the plasmid or extra chromosomal resistance. Okay, so MRSA, so methicillin-resistant Staph aureus, is an example of a plasmid-based resistance. Okay, so lots of drugs that are out there don't work anymore against certain organisms. Okay, so some abbreviations. So MDR is multi-drug resistant, and then XDR is extreme drug resistance. And this is Mycobacterium tuberculosis. So that is what causes tuberculosis, or TB, which is a lung infection. It's pretty contagious. It stays around for a long time. I mean, I guess it's not super contagious, but it's hard to treat. Mycobacterium grows really, really slow. And it's not remember, it's not a gram, it's an acid fast organism, so the antibiotics don't necessarily work on it the same way it would if it was like staph or E. coli or something like that. So it takes a really long course of antibiotics to get rid of it. So what they do is they'll actually give a combination of drugs, so like isoniazid and rifampin together, or fluoroquinolone, which is Cipro, and something else. Okay, so they've burned through a lot of drugs. And if you think about it, okay, if it takes a long time to treat it, so if it takes 6 months to treat it, and so you have to take this antibiotic for 6 months, right? They don't stick with it. They don't keep taking it. Okay, now MRSA, okay, is methicillin-resistant staph, okay, and then there's other ones. There's a vancomycin-resistant enterococci, or ones that are found in the gut. A lot of things come from the hospital, so they're nosocomial infections. So, and we talk about those more. So nosocomial infections are infections that come from the hospital. So they've been exposed to more antibiotics, so lots of times they're more sensitive. Gonorrhea is kind of crazy because it's the second highest infectious disease reported in the U.S., so this STI, and we've used a lot of antibiotics, and there's a lot of antibiotic resistance to it. So it's kind of crazy. Okay, so mechanisms of resistance. This numbered it really weird, but there's 5 of them. So there's 1, 2, 3, 4, 5. I don't know what happened when I imported it. Okay, so the first one is that a receptor on the bacterium has been altered so that the antibiotic doesn't bind to it anymore. Okay, sometimes the bacteria will alter their membrane permeability, so the antibiotic can't, or the antimicrobial can't enter the cell. So with that, we could add something like penicillin to it, and then we could damage the cell wall and then break it down. Okay, sometimes they develop new enzymes. So with penicillin and cephalosporin, the active part of the antibiotic is this beta-lactam ring. So bacteria have developed this enzyme called beta-lactamase, and beta-lactamase breaks down this beta-lactam ring, and it makes penicillin not work anymore. Okay, they could alter an enzyme. So what they do is they become better at binding something like PABA, so then that gets rid of the competitive inhibition, or they develop totally new metabolic capabilities. So bacteria, they're constantly changing in response to their environment. Okay, so here is a picture, and I just wanted you guys to be able to see. Okay, so this blue thing is the beta-lactam ring, and that's what interferes with the peptidoglycan and makes penicillin and cephalosporin work. But bacteria have developed this enzyme beta-lactamase, and that breaks down this ring. So then the antibiotic doesn't work anymore. Okay, so the ways that we try to limit drug resistance is we try to control the environment. So we want to give a high enough dose, and you need to take the whole dose. So you take it long enough to kill or destroy the bacteria. So here's an example. This isn't very big, it's hard to see, but here's the idea. So day 0, these are the organisms in the body. Okay, and then day 3, we've started to select towards the ones that are resistant. And then day 6, we are still trying to kill the resistant ones, and after day 10, we've reduced the number of resistant ones by a lot. So if you stop when you start to feel better, okay, so like a weekend, you're like, oh, I don't need the last couple days. So then what happens is these guys, right, they survive, and they start growing. So then the next time you get sick, you have these highly resistant ones in there, or you have a relapse. Okay, so you got to take the full dose. Sometimes what we do to try to reduce resistance is give 2 antibiotics together, or we modify an antibiotic so that it works more effectively. Okay, so streptomycin and penicillin we put together. Augmentin is amoxicillin, so a penicillin, and they've added this clavulanic acid to it. That's pretty common. Okay, and then the last thing is to only prescribe antibiotics when it's necessary. So like somebody says they have the flu and they gave them an antibiotic. Okay, remember, flu is caused by a virus, so the antibiotic isn't going to do any good. So don't take them unless you have to. Okay, now, one way that they're trying to develop a new way to control bacteria is the idea called quorum sensing. So a quorum is having a certain number of, okay, so a quorum is, the idea is that you have a certain number of organisms. So like if you're voting, like if you're Congress or you're an assembly, in order for a law or a law to pass, you have to have a certain number of members there present and voting in order for it to actually count, for the vote to count. So what happens is some bacteria, you have to have a certain number of them accumulated in order for them to start talking to each other enough to produce a toxin. Okay, so 2 examples of organisms. One is Vibrio cholera, which causes cholera, which is a really bad diarrhea. And then the other one is E. coli, and it's 157H7. So this is the E. coli and the strain of E. coli that causes bloody diarrhea, like the Jack-in-the-Box kind of diarrhea. Okay, so what happens is, what they're trying to figure out is if they can let the bacteria live, but if they shut down the way that they communicate with each other. So if you have this whole bunch of bacteria, Okay, so you have a whole bunch of them, and they have to talk to each other so they know that they're all there. Okay, once they know they're all there, then they'll start making the toxin. So if we can go in and we can shut down communication, okay, so if you can stop communication, they'll never know the other ones are there and they won't make the toxin. So these molecules, the red things, the molecules that they're communicating with are called quorum sensing inducer molecules. Okay, so if they can shut those down somehow, okay, you could have the organism there, but it's not going to produce the bad thing. Okay, so far they found 20 drugs that interfere with this inducer molecule, and they're trying to figure out how to use them. So that's new. That's kind of new, but it's out there. Okay, now 3 ways to prescribe antibiotics. So one is prophylactically. So prophylactically means you're using it before you get sick. So to prevent symptoms. So one example of that would be people that have heart valve problems or heart murmur. Lots of times before they go to the dentist to get their teeth cleaned, they get a dose of penicillin. Okay, because if they're scraping the bacteria off your teeth and it gets into your bloodstream, the bacteria can actually accumulate in the valves. So they try to head that off by prophylactically. Okay, now empiric is based on the symptoms, and you don't know for sure what infection they have, so they kind of call it the best guess. So they're going to look at what's going on, or look at the symptoms. They're going to look at what's in the environment, where you're coming from, and try to treat that way. And then the best ideal way to do it, which is not always possible, is to do a swab, identify it, and eliminate what's actually causing the problem. Okay, so how are they administered? So there's oral, there's intramuscular, there's intravenous. Okay, so oral, it has to get into the GI tract and to the liver and to the blood, and then it goes to the target. It takes like 30 to 60 minutes to work. You have to remember every drug has a half-life. Okay, so it only stays in the body for a certain period of time. Okay, IM or intramuscular, it's going straight into the muscle. So it's going to work a lot faster, but if the drug isn't water soluble, it's harder to get it into the muscle. So it's going to work faster and you get more in. And then IV, it works really, really fast. Oops, wait a minute. Okay, now to determine what drugs to use, one way is with this Kirby-Bauer plate. So this we did in lab. So you measure the diameter. It all depends on the diffusion of the drug. It's definitely standardized. Some things that could happen is that it might not work in people if you can't get the antibiotic into the blood. And another thing is that it could be static and not cidal. But it's a way to assess a bunch of drugs at one time. It's really simple, it's cheap. But if the organism's hard to grow or it's an anaerobe, that's going to make it trickier. Okay, when you interpret the Kirby-Bauer test, what you do is you just measure the zone of inhibition in millimeters, and then you determine whether it's sensitive, intermediate, or resistant. So if it's sensitive, the doctor can give it orally. If it's intermediate, you probably don't want to give it orally. You won't be able to get enough drug in, but you can do IM or IV. And if it's resistant, it's not worth it to try that drug. Okay, now an epsilometer test is a way to tell how much of the drug you need to be effective against the organism. So instead of having a whole bunch of discs, what we have is we have a strip. We have a strip. And the high concentration is out towards the edge of the Petri plate, and the lower concentration is in the middle. So basically what happens is you make a lawn again, and you put the strips down, and you look, this is the zone of inhibition. So you get it kind of like an egg, And at the bottom of the zone, that's where the minimum inhibitory concentration is going to be. Okay, now this is even more accurate. So what they're going to do is you're going to use a plate. So usually these have 96 wells in them, and you dilute the antibiotic. So we're going to increase the dilution of the antibiotic, and then you add the bacteria, and you put it in 37 degrees, and you allow it to grow, and then you can look at the plate, and you can determine if there is growth there or not. So this test can be automated, and it's a way to determine the minimum inhibitory amount of antibiotic that's necessary. This is a way that they screen for new drugs too. Okay, serum killing power. I don't have a picture of it, but the idea behind this is that this is in people, right? So you're working in a person, so you're going to give a patient the antibiotic. You're going to take a blood sample, and you're going to look at the serum and see if there's enough antibiotic in the serum to actually kill the bacteria. So this would be done like in clinical trials. So if they start out with healthy people, okay, and they give them the antibiotic, and then they take their blood, separate out the serum, and then see if they could get the organism to grow. Okay, so here's 8 ideal drug attributes. So it should be soluble in body fluids, shouldn't be toxic to the host, okay, shouldn't interact with stuff, like it shouldn't interact with your food or other drugs, okay, shouldn't cause allergic reaction, it should be easy to maintain a constant level in the body, resistance should be acquired slowly, long shelf life. Okay, so, and if you don't have to keep it cold, that's even better. And then the last thing is a reasonable cost, which is kind of crazy right now, especially with new drugs.